Thanks, Emma, for that nice introduction, and thanks to the organizers for inviting me to give this talk today. As Emma told you, I'm actually a geneticist here in Seattle at the Pacific Northwest Research Institute, and today I'm going to tell you about one of the microbes that my lab works on, a study that we did to actually examine the microbes that are associated with green coffee beans isolated from around the world, and some surprising results that we had that might change how you think about the fermentation of coffee. But first, let's just start with microbes. Microbes are everywhere, and by microbes, I mean bacteria, yeast, and molds. And these are present all over the outside and inside of our bodies. You may have heard this called the microbiome, and these microbes actually perform vital functions that are essential for human health. But of course, these microbes also exist all over the outside, inside of all other organisms, as well as throughout the environment. And these microbes aren't just hanging out doing nothing. They're performing vital processes that are constantly modifying the environment that's around them. So today, before we get started, I'd like to introduce you to my favorite microbe, the yeast Saccharomyces cerevisiae, which most of you know as baker's yeast. Now, if you're the sort of person who also has a favorite microbe, cerevisiae might be one of your favorites as well. And that's because baker's yeast is involved in the production of many of the foods and beverages that we all know and love. Of course, almost everyone knows that baker's yeast is involved in the production of bread, beer, and wine. And today, I'm going to tell you about its involvement with a product that I know you all care about, which is coffee. But first, I'd like to tell you a little bit about something most people don't know about baker's yeast. And that's the fact that yeast is actually one of the most intensively studied organisms used in biomedical research, which is, of course, what my lab does. In fact, several Nobel Prizes have actually been won for studies that involve baker's yeast. One of those was for research that was performed right here down the street by Dr. Leland Hartwell. Dr. Hartwell is a yeast geneticist who did his prize-winning research at the University of Washington. And he made some amazing discoveries about how yeast cells grow and divide. And because these same processes allow human cells to grow and divide, understanding these processes is important for preventing and treating cancer. In fact, just this year, the Nobel Prize in Medicine and Physiology was won by another yeast researcher, Dr. Yoshinori Osumi. Dr. Osumi's research focused on how yeast cells degrade and recycle some of their cellular components. And again, because these same processes allow human cells to degrade and recycle their components, these processes are important in combating infection and preventing neurological diseases. So these are two amazing studies that illustrate why researchers like the ones in my lab study yeast as a model system. And that's because yeast cells are incredibly similar to human cells. In fact, they're so similar that we can actually take human genes, put them into baker's yeast, and they'll usually perform almost the same function. But as similar as yeast cells are to human cells, they're even more similar to the cells of other fungi. And those include pathogenic fungi that can affect premature infants and people whose immune systems are compromised by disease or chemotherapy. And so my lab studies baker's yeast for both of these reasons, to understand how human genes function and help prevent and cure human disease, and to understand how pathogenic fungi respond and develop new antifungal drugs. So one of the things that my lab needs in the course of our biomedical research is a large and diverse collection of very diverse yeast. Now, I know that the coffee community is keenly aware of the need for genetic diversity, but it may surprise you to know that even in the biomedical research field, we're sometimes limited by genetic diversity. When my lab started the project that I'm about to tell you about, the number of yeast strains that we had available to us was very small, and they were all very similar to each other. And in my world, that's the equivalent of trying to study how different people respond to, for example, a drug treatment by only studying you and your identical twin. So we needed a large collection of genetically diverse yeast, but we couldn't just go to the store and buy them. And so, of course, one way to find new and exotic organisms is by looking in new and exotic places. That is, to look around the world, and especially in places where people hadn't looked for yeast before, and try and find new sources. So we decided to go on a sort of worldwide microbe safari. And of course, one very pleasant way to do this would be to visit vineyards, because wine is made around the world, and yeast is involved in the production of wine. Now, when vintners add yeast to crushed grapes in order to make wine, the yeast not only produces alcohol from the sugars that are in the grapes, but it also imparts volatile organic compounds that give the wine its flavor and aroma that it's very pleasant in some cases. And one thing that you may or may not know is actually these volatile compounds are actually produced by the yeast themselves. And so it's also the case that different genetically diverse yeasts will make different compounds under different environmental conditions and different situations. And so in the plot I have here, this is a graph from one of my collaborators, Dr. Justin Fay at WashU in St. Louis. And what Justin's lab did was to take two different yeast strains. One is a strain that's commonly used in wine, the strain here in red, and another is a wild strain that they isolated from an oak forest. And then they took sterile grape juice and produced wine from each of these, with each of these two yeast strains. And you can see here that when they had these wine samples professionally tasted by professional wine tasters, that the wine strain produces aromatics that are reminiscent of floral and citrus flavors, but the wild yeast actually produced essence of cabbage, mushroom, and wet dog. And I certainly know which one of these I'd like to drink. But unfortunately for my dream of visiting vineyards throughout the world, researchers already knew that actually yeast strains isolated from different vineyards around the world were incredibly similar to each other. And one of the reasons why this is thought to be the case is that winemaking was actually perfected very early in human history, before humans began large-scale migrations. And one of the things that early civilizations learned about winemaking is that they could actually get better, more reproducible wine by starting a new wine fermentation with the remnants of a previous fermentation. And by doing this over and over again, they selected or domesticated strains of yeast that are actually really good for winemaking, such as the one that I showed you there. And so then, as humans did start to migrate throughout the world, they not only brought with them the crops, the grapes, but they also brought with them the vessels and these starter cultures for making good wine. And as a result, the yeast that anyone samples from vineyards in France, Australia, South Africa, California, or even here in Washington State are genetically very similar to each other. And that's the case whether or not you isolate the yeast actually from the wine itself, from the grape skins, or even the soil on the floor of the vineyard. So unfortunately for our microbe safari, visiting vineyards throughout the world was not going to help us. But as we were thinking about how to address this problem, it occurred to us that we knew of two other crops that were both grown in the types of exotic places that we were interested in surveying, and were also the result of fermentation. And those are, of course, cacao and coffee. Now, you all know that both cacao and coffee are fermented in country, and then after light processing, the unroasted beans can be transported throughout the world. And in the case of cacao, it was well known that their yeast, or that baker's yeast, is actually an important component of those fermentations. However, at the time we started our studies, there was very little evidence, actually, that yeast was even involved in the production of the fermentations of coffee. But we reasoned that if there were actually live yeast still stuck to these unroasted beans, cacao and coffee, that we should be able to isolate them back in our lab here in Seattle, Washington. So let me first start by talking about cacao. Raw cacao beans are unedible, and I've been told they taste terrible. And that's why, in the production of chocolate, farmers hack open the pods with a machete, scoop out the mucilaginous pulp, which also contains the beans, and heap them either into wooden boxes or onto banana leaves on the forest floor, where they oversee a 1 to 2 week fermentation process. Now, scientists know a lot about these fermentation processes. One of the things they know is that they're very complex. They involve a large number of diverse microbes, so yeasts, bacteria, and molds. The second thing that they know is that these fermentations occur in an ordered fashion and they're very reproducible. That is, certain microbes appear in the fermentation and then other microbes appear and take over. And one of the reasons for that is that each of these microbes are using up and then producing different metabolites. So for example, yeast will utilize sugars and produce ethanol. That ethanol can then be utilized by a different microbe. And in these ways, different microbes proceed through the fermentation in succession. The final thing that they know is that these aren't just using up sugar and producing ethanol and degrading the pulp, but these microbes are actually imparting volatile organic compounds, like the ones I talked about in wine. And yeast especially are involved in the production of these volatile organic compounds, which change the color, the taste, and the aroma of the chocolate. And in fact, in the case of cacao, recent studies have shown that yeast are very important for imparting these flavors to the chocolate. And in fact, that if you inoculate the fermentation, you spike the fermentation with specific strains of yeast, you can isolate chocolate with more favorable properties, so increased aromas of bitterness, fruitiness, and cacao. So what's known about coffee? In the case of coffee, as I said, these fermentations are not well understood, nothing like the case of wine and cacao. We don't know what microbes are present in the fermentations. We don't know what compounds they might be utilizing and producing. And we don't even know how important these microbes may or may not be for the final product in the cup. But there we are, and so let's get back to my microbe safari. So in our efforts to isolate genetically diverse yeasts, one of the things that my lab did was to develop a rapid method for isolating yeast from unroasted cacao and green coffee beans. And to do that, what we do is we take a few green coffee beans, we place them into a sterile test tube with some growth media. So this is basically liquid that contains all of the sugars and nutrients that the yeast cells need to grow and divide. And so if there are live yeast present on the green coffee beans, what we'll see is that those cells will start to grow and divide, and as a result, that liquid will start to become cloudy, and it will start to bubble as the yeast start to ferment. Now, I should tell you that this growth media that we're using is basically glorified sugar water. It has very little scent to it. And so I knew that we were really on the right track when one of the scientists in my lab, Kathy Ludlow, called me over to her lab bench, pointed to one of the test tubes and said, "Smell that one." Now, I have an 8-year-old son, so anytime someone tells me to smell something, I'm very suspicious. But I trust Kathy. So I opened the tube, and I was delighted to find the most delicious aroma of chocolate coming from this sugar water with coffee beans in it. And so Kathy explained to me, actually, that all of the different test tubes she had on her bench with different samples of coffee beans were actually producing sometimes subtly different aromas and sometimes drastically different aromas. Sometimes they were lovely, and sometimes, to my nose, they were not very pleasant. But it was almost certainly the yeast and maybe other microbes that were associated with these beans that were producing those. And so this bode really well, we thought, for the potential for isolating genetically diverse microbes. But were we able to actually isolate yeast from these? And the answer is happily yes. In fact, we were very successful at it. So from a relatively limited sampling of, again, coffee and cacao, we were able to isolate 140 genetically diverse yeast strains. And here is a list of the countries that we were successful at isolating from in the case of coffee. But But were these like the wine case, where even though these came from sources all over the world, they were actually genetically very similar, or were they more genetically diverse? To answer that question, we took each of these 140 yeast strains, together with a collection of another 400 strains that we had collected from other sources, and we read the same 3% of their genome, their DNA sequences. And here on this graph is one type of depiction of the DNA sequences of those yeast strains. So here, what you're seeing, every row of this graph is one of those 540 yeast strains that I told you about, and every column is a position in their DNA sequence. And we've colored those positions black if they have a DNA sequence that's common in the population, red if they have a rare sequence, and yellow if they have one of each. So as you can imagine, these are big data sets, and we analyze them with rather intricate statistical methods. But let's start by just asking a very simple question. Are the yeast strains that are associated with coffee and chocolate genetically similar or more diverse? So now I'm going to show you a different way to represent this data. And now, on this type of plot, every dot on this plot is a different yeast strain that was either isolated from vineyards, coffee, or cacao. And so, the other thing you need to know about these plots is that, so every dot is a yeast strain, and the closer they are together, the more similar their DNA sequences are to each other. The farther apart they are, the more different their DNA sequences are. And so one of the things that I hope will be really obvious is that you can see these blue wine strains that we have are all bunched up together. And the reason for that is that their DNA sequences are very similar to each other. However, one of the things I hope you'll also appreciate is that the coffee and cacao strains are much more genetically diverse, both from the wine strains in many cases and from each other. So this was really exciting because it said that we have our collection of genetically diverse yeasts that we wanted for our biomedical research. So we were very happy. But one of the other things we noticed when we looked at the data more closely was that actually the DNA sequences of these yeast strains grouped together in another kind of way. And that is that yeast strains grouped by both the source, coffee or chocolate, as well as geography. In other words, yeast strains from Colombian coffee were very similar to other yeast strains from Colombian coffee, but distinct from coffee strains from Yemen. And this was true regardless of the supplier that we had, and we did this multiple times to test this. And that DNA signature is actually so clear that if you hand me a green coffee bean, I can tell you where it was grown and fermented based solely on the DNA sequences of the live yeast that I can actually isolate from it. The other reason why that's exciting is because what this is suggesting is that there are local microbes associated with places where coffee and cacao are being grown, and that these microbes are different no matter where, depending on where you go, depending on where the crops are grown and fermented. And the reason why I think that's important for the coffee industry is because, remember these wine strains that we have up here that are all bunched together in blue? They're very similar to each other. But research has shown that, that a small but significant amount of the terroir associated with wine processing actually results from the microbes themselves. And so if you can have differences in terroir that are resulting from strains that are this similar, imagine the differences that might exist between the genetic diversity of the coffee and chocolate yeasts that we've shown in our study. So, given the results of our research, what advice would I give to the coffee industry? Because I'm not a coffee researcher. So the first is that I think it's pretty clear that it's important to understand these microbial fermentations. And at the very least, understanding, like the case in wine and to some extent in cacao, understanding what is involved in a successful fermentation could help prevent loss due to waste, due to poor fermentations. The second is that I've shown you today that our research has demonstrated that these yeasts are very genetically diverse. And so, and that also this diversity is localized to different regions in the globe where coffee is grown. And so the potential, as I said before in my introduction, we don't know what the influence of these microbes on coffee production even is. But I think it's really important to try and understand this because this effect on terroir could be pretty substantial. And if it is substantial, there's a third possibility, which is that inoculating coffee fermentations, as you saw in that nice video that Emma showed, has the potential to either give you a more controlled product or a vastly different product, depending on which yeast strains you might add either alone or in combination with other microbes. And finally, if you don't remember anything else that I've told you today, I'd like you to take home this one point. Biomedical research, like the type that's performed in my lab, very often leads to new, exciting, and completely unpredictable directions. And so if your main concern is improving the discoveries that will improve your business or the economic development of the region that you care about, supporting biomedical research, basic research, with your tax dollars and your philanthropy is one of the most important things you can do. Thank you very much.